My name is Meera and I'll be a presenter today for the topic Applications of Biotechnology in Medicine. But before seeing the applications of biotechnology, let us see what biotechnology is. So biotechnology is the techniques of using live organisms or their components or enzymes derived from them to create products or processes that are useful to us humans. So biotechnology is carried out in three main steps. Creation of recombinant DNA, where we isolate the gene of interest and the vector, it could be a plasmid or a bacteriophage. They are cut using the same restriction endonuclease and ligated using the enzyme DNA ligase. So that creates the recombinant DNA. This recombinant DNA is then cloned and then transferred into the host cell or the host organism. So biotechnology, knowingly or unknowingly, we have been using it since ancient times. For example, in the making of cheese or curd. While making curd, we just add a small amount of curd into the milk. We don't have to do anything additionally for it, right? So the bacteria present in that small amount of curd itself does all the work. And it is the modern applications of biotechnology that helps us in making DNA vaccines and helps us carry out IVF. So biotechnology has its applications in agriculture, food processing, waste remediation, therapeutics and medicine. So what is medical biotechnology? Medical biotechnology just means the use of biotechnology in medicine. We use biotechnology to do research and then find or invent new products and therapeutics that can be used to treat and prevent diseases. The first application of biotechnology in medicine is genetically engineered insulin. So we know what insulin is. Insulin is a hormone that is released in our body by the pancreas and it helps in controlling the blood sugar. So more people are developing diabetes. So there's a greater demand of insulin all over the world. Where do we get this insulin from? Earlier, insulin was extracted from the pancreas of slaughtered cattle and pigs. And though this insulin is not very chemically different to the insulin that is produced in our body, but still people face allergic reactions. Because our immune system, it would reject anything that has come from an outside source. So this won't work. So we cannot use this insulin. We need some other source of insulin. So before understanding that source, we'll see the structure of insulin first. So insulin has three polypeptide chains: chain C, chain A, and chain B. This chain A and chain B are connected by disulfide bonds. So this is how it would look. This is the chain B, the longer chain, polypeptide chain B. This is the chain A and this is the chain B. This is the chain C. Chain C, chain B and chain A, and these are the disulfide bonds by which the chain A and chain B is connected. So this is the proinsulin. Insulin in our body is released as proinsulin that is not used by our body. Proinsulin should undergo processing to form the insulin that is used by our body. So the insulin after processing does not have the C chain. It only has the A chains and the B chains that are connected by disulfide bonds. So this is the insulin that is used by our body. So the insulin used by our body that is needed to control the blood sugar has a chain B and chain A that is connected by disulfide bonds. So this is the insulin that is needed by diabetic patients because they don't have this insulin in their body. So in 1983, an American company, Eli Lilly, artificially created human insulin by preparing this human insulin, in a lab. They made it. How? No one knows. I'll see. So they prepared the DNA sequences that codes for chain A and chain B and introduced it into the plasmid of E. coli bacteria separately. They take the chain, this is the DNA sequence for chain A. They introduce it into the plasmid of E. coli bacteria. This is the DNA sequence that codes for chain B and they introduce it into the plasmid of E. coli bacteria. And this bacteria is allowed to multiply and these two chains are extracted and purified. And they are connected by creating disulfide bonds. And human insulin is formed. That is used by people. Inject. I'll show a video. Insulin is produced by the pancreas. We all know that, okay? translation into nucleus. This is the gene that we need to produce the insulin. This is the vector. It is a plasmid of the E. coli bacteria. And this gene that codes for insulin and the plasmid is ligated to form the recombinant DNA. This is introduced into the E. coli bacteria and is allowed to multiply. Next is gene therapy. Might seem difficult in the first go, but it's easy, very easy. Gene therapy is a collection of methods that helps us to correct a defective gene. So it is done by inserting the correct gene into the cells or tissues of the patient. So this helps us, gene therapy helps us to solve the problem at the gene level. In gene therapy's first clinical trial was in 1990 on a 4-year-old girl who suffered from ADA deficiency. ADA stands for adenosine deaminase. So why do we need this ADA? This ADA is an enzyme that is produced in our body that supports the immune system. So in this girl who is suffering from ADA deficiency, her immune system would be very weak. So she'd be more vulnerable to diseases. So before gene therapy, these were the two treatments used to treat ADA deficiency. Enzyme replacement therapy means you just inject the ADA enzyme into your body, just like we inject insulin. So, but this is not a curative measure. Why? Because ADA is continuously used by our body to support the immune system. So we need to. So after like 5 minutes, we have to inject it again and again. So this is not a curative measure. Next is bone marrow transplantation, where the lymphocytes. See, the main problem is that in this girl, the lymphocytes does not have the gene that encodes for ADA. We have to correct that. So in bone marrow transplantation, the lymphocytes that has the ADA gene, the gene that produces the ADA enzyme, is transfused into her body. But see, not everything is not mortal. Sorry, immortal. I'm sorry. Everything is mortal. So it has to be. It requires periodic transfusion. Like after 3 months, these lymphocytes should be injected into her body again and again. So, as the first step towards gene therapy, the lymphocytes of the patient, of the girl, is taken out. Like a few of the lymphocytes is taken out, and it is grown in a culture outside the body. And this gene, sorry. Now, and this, see, this is the lymphocyte. Her lymphocyte does not have the ADA gene that codes for the enzyme. Okay. So into this lymphocyte, we add the gene. We add the gene that encodes for ADA enzyme, and then there is the gene, it produces AD enzyme. Finish. Simple. Understood? Understood?
Yes.
Sir?
Yes.
Thank you. Not yeah. So, recap. This girl has ADA deficiency, meaning her lymphocytes does not produce ADA enzyme. So, this is her lymphocyte. We take it out of her body and it is grown in a culture outside the body. And then the gene that encodes for the ADA enzyme is introduced into her lymphocytes. These lymphocytes are then introduced back into her body. This also we'll have to do the periodic transfusion because she's 4-year-old. But gene therapy can be a permanent cure if it is done at the early embryonic stage, like at the 2-cell or 3-cell stage. But it is easier because the number of cells is very minimum. Another video. Very nice video.
Genes make us who we are. Are we tall or short? Do we have blue eyes or brown? Our genes hold the answer. What if part of a gene is defective or missing? Or if a gene changes or mutates? It can cause a disease like cancer. Enter gene therapy. Among other things, scientists can replace a bad gene with one that is healthy, add a new gene to perform the function of a defective or missing gene, or they can turn off a problem gene. How do they do it? Say scientists want to add back a healthy copy of a gene to cells. They need an efficient way to do it. So scientists use vectors or vehicles to deliver a gene. The healthy gene hitches a ride in the vector. Viruses are one kind of vector. Scientists modify a virus to remove its ability to cause a disease. The modified virus can then be used to carry the healthy gene into a cell. Gene therapy can happen on cells inside the body, or can happen on cells outside, and then transplanted back in again. Genes make us who we are, and gene therapy holds great promise for a healthy future.
Next is molecular diagnosis. Molecular diagnosis means different tests to diagnose the diseases at an early stage. Because normally by the time we suspect something is wrong, the concentration of the pathogen in our body would be very high. So PCR test is the first one. PCR stands for polymerase chain reaction. It is called a chain reaction because in this PCR test, what we do is that we amplify the nucleic acid. We amplify the genome of the pathogen that has entered our body. So the one DNA becomes 2, 2 becomes 4, 4 becomes 8. It is multiplied multiple times, and then we get an idea, oh, something is wrong. This person has the genome of this pathogen. So if we treat immediately, it will be done. He can be treated. Next is the ELISA test. ELISA stands for enzyme linked immunosorbent assay, and it works on the principle of antigen antibody interaction. So what is an antigen? Antigen is a foreign body or foreign substance that enters our body, and antibodies are the proteinaceous substances that is produced against this antigen by our immune system. So it helps us by detecting the presence of the antigen in our body, or by checking if some pathogen has entered my body. If I take a blood test, the antibodies level will be very high. So that tells us that something is wrong. Some antigen has entered our body. So it is very helpful in diagnosing diseases. Next is autoradiography. Autoradiography, a probe is used. A probe is a single-stranded DNA or RNA that is bound to a radioactive molecule. A single-stranded DNA or RNA that is attached to a radioactive molecule. Now this probe is allowed to hybridize to its complementary DNA and shown on a photographic film. And if there is a mutated gene, this can be used in cancer patients, because in cancer patients most of the times it is because of mutation of genes that it happens. So when this complementary DNA is shown on a photographic film, the mutated gene would not appear because this probe cannot form its complementarity to the mutated gene. Wow, another video. Next is genetically engineered vaccine. What is a genetic vaccine? A genetic vaccine is a vaccine that has the genetic material or nucleic acid of a DNA of a pathogen. So there's a vaccine, it has the genetic material of a pathogen. So if I inject it into my body, my immune system reacts against it. It produces the antibodies that is needed to fight this pathogen. So if in actual this pathogen enters my body would recognize, yes, this is that same intruder. I need to produce the same type of antibodies against this. So our body produces antibodies in more number. So that is how we can prevent diseases. Next is a recombinant vaccine. Recombinant vaccine is prepared using recombinant DNA technology. So recombinant DNA technology and biotechnology is same thing. So in recombinant vaccine, we insert the DNA encoding an antigen such as this protein found on the surface of the pathogen like a bacteria, and this stimulates an immune response. Same thing here. When we inject this thing, this vaccine has the genetic material of the pathogen. This vaccine has the protein or the surface antigen of that pathogen. So when this is injected into the body, our immune system produces antibodies against it. So that prepares our immune system for this attack. The first such recombinant vaccine was against hepatitis B, where the recombinant DNA produced hepatitis B surface antigen, which is made by the yeast Saccharomyces cerevisiae. And another one is HPV, human papilloma virus, which has shown to prevent most of cervical cancers and genital warts. Last video.
The concept of recombinant DNA technology is to harness the manufacturing power of the world's smallest factories, living cells. Organisms like bacteria and yeast are able to share genes by trading rings of DNA called plasmids. These plasmids carry the blueprints that instruct cells to make specific proteins, like the protein Maurice Hilleman used to make his hepatitis B vaccine. By using a special enzyme to cut the plasmids, scientists can insert a gene that instructs the cell to make the hepatitis B surface protein. Vaccines
This DNA encodes for the antigen, the surface antigen.
Easier to make, easier to purify, and just as safe as live or killed vaccines.
Thank you. Any doubts?
Let's do now question answer section.
No doubts?
Any
Can
question?
you explain genetically engineered part again? Because it wasn't pretty clear.
Genetically engineered vaccine? Genetically engineered vaccine. So genetically engineered vaccine, this is not genetically engineered, this is what is genetically engineered. So in this, the surface, the protein or the antigen that is on the surface of the pathogen is like we prepare a gene that encodes for the surface antigen. Okay? And then that is ligated with a plasmid that is allowed to multiply, and that is made into a vaccine form. And when we inject this vaccine into our body, our immune system reacts against it by producing antibodies against this pathogen's surface antigen. So when this pathogen actually enters our body, our immune system knows that this pathogen has entered our body before, this is not good for us. So we have to produce the same type of antibodies in larger amounts. Any other doubts?
Could you explain the process of ELISA again?
Repeat the question.
Yes. The process of ELISA, like ELISA works on the principle of antigen-antibody interaction. So when, you know what an antigen is. So when an antigen enters our body, antibodies are produced against this. So like if we take a blood test, we would know that if some antigen has entered our body, when we take a blood test, the antibody level in our body would be very high. So that would help us know that there is something wrong, like some pathogen has entered. So this would help us in diagnosing the disease before the concentration of the pathogen has become very high in our body.
Is there any particular disease that this test is for?
Like maybe like HIV, because in HIV and all, HIV, AIDS and all, the symptoms come after like a very long time. So it can be used in such this.
But even PCR does the same thing. It recognizes it when
Yes, PCR also does the same, but see, we cannot, not everything is 100% efficient. We need to, like in AIDS tests and cancer tests, we combine different methods of tests. Like for diagnosis, different tests are used so that we're like sure. So both of them can be used. Any other doubts? No? Oh, yes.
When you were talking about insulin, you were talking about restriction endonucleases, right? Is there a specific type of restriction?
No. No, it's there, but I don't know what it is. I'm sorry. I think it is there.
Go on, children. Thank you.
Thank you, ma'am.